Now we will continue our discussion of different types of markers, in this video focusing on single gene sequencing. And we've actually talked a little bit about this before. In the previous module, we talked about PCR and Sanger sequencing, which is essentially how you get single gene sequences to use as markers. So we have some prior background knowledge about this. And in this question, I'm asking you to think about how is DNA sequencing different from microsatellites, which we just talked about in the last video. So I'll give you some time to think about this first on your own. You can jot notes on the slides if you have those printed out or just in a notebook. How is DNA sequencing different from microsatellites? One of the main differences is the types of data that you get. So DNA sequencing, what you're interested in, which is shown here on the bottom in these chromatogram images, are the actual nucleotide differences. NT is often an abbreviation for nucleotide. So when we're using DNA sequencing, in this case from a single gene as a molecular marker, really what we're looking at are these actual nucleotide differences. So that's indicated by this black arrow here. Those are actual nucleotide differences between individual A and individual B, for instance. With microsatellites, in contrast, really what we're looking at and are interested in is the differences in the number of repeats. So like we said when we talked about this in the previous video, that's how microsatellites differ from each other. It's not in their sequence. So if we're talking about this locus 2 microsatellite, everything is a repeat of TAA. So all the nucleotides are the same. What's different is how many repeats there are. So this one has 8, this one has 7. So that's what we're interested in. Microsatellites can give us some information about differences between individuals if they have a different number of repeats for that particular microsatellite, for instance, but they cannot give us any information about actual nucleotide differences. That's something that only DNA sequencing can give us. And as we talked about in the previous module, PCR, which is shown here on the left, we have our template DNA, our primers, and our new synthesized DNA. That process of PCR, which is then coupled with Sanger sequencing that produces these chromatogram images on the right, that process is what produces these single gene sequences that we can then use as molecular markers in molecular ecology studies. And one really important consideration, well, I'm going to ask you what you think that important consideration is. Based on what we've talked about before and what I just showed on the last slide, what decides what region of the genome we amplify through PCR, and therefore what region we will sequence and obtain that DNA sequence from? What do you think the main factor is? So what really decides what region we amplify is our primers. So the primers are what specifically bind to our target template DNA and then amplify that region that we want to copy. So the design of the primers and the specific primers we use really what determines what sequence of the genome we're able to obtain. And there are two different general types that we'll talk about in this video. And we'll start with universal PCR primers. Universal primers are called universal because what they do is amplify the same region of DNA from different species. So what I have here in this image is an alignment of a DNA sequence from very different types of species. So goat, sheep, deer, buffalo, cattle, yak, pig, and camel. And what's shown here in the blue boxes are regions where universal primers can be designed to bind to, because even though those species are very different, that particular region, the nucleotide sequence is the same. Mostly the same. I see some actual differences here, but for the most part, you can design primers that will bind to any of those species that are listed there. And that's beneficial if you're starting work with a new project, a new species, for instance, and you don't know a lot of information yet about that particular species genome. You can use universal primers that have been designed for a larger group of species, and they'll also work on the species that you're interested in. The other type of primer is a species-specific primer. And as the name suggests, what these primers do are they specifically target, and they only amplify target sequences from one or a small number of target species. So here what I'm showing is an alignment of DNA sequences from 5 different freshwater mussel species. And in bold here, I'll use blue. This is a region of that gene that is conserved. It means it's the same for all of those species. So this is where we would design a universal primer. If we had a universal primer, that would work for all 5 of these species, because this sequence here in this region is exactly the same across all 5 species. But if we wanted to use a species-specific primer and only target one of those species, we would want to look for regions in this DNA alignment where the sequences are different, or they're not the same among the species. So in this top row here, each one of these little asterisk stars indicates that in the nucleotide directly below that asterisk, that all of those nucleotides are the same. So if you look, even though these are different species, there are a lot of asterisks, that's a hard word to say a lot of times, on top of that sequence. And if we want to design species-specific primers, what we want to look for are regions where we don't have asterisks, because that means there are actually differences among the different species. So I have highlighted in that blue box where we would want to design a universal primer. But if we wanted to design a species-specific primer, we might want to do it somewhere like in this region where we have several different non-asterisk parts, which means that there are actually nucleotide differences that we would be able to use to design a PCR primer that would only bind to the one species that we're interested in. A PCR primer is only going to bind to a DNA template if it's a good match. And if we design a PCR primer to only bind to that one species we're interested in, that means the PCR primer will only produce a PCR product for that species that we're interested in. We can then sequence that product. That primer will not bind to any other species though, so we will not get any other PCR product or data for the other species, which is what we want in this particular scenario. So that differentiates universal and species-specific primers, which are an integral part of the study design for single gene sequencing.